All right, so as I just reiterated to you guys, these problems, give these a try, right? 14, you should be able to do easily. 15, try it. 28 should be pretty easy. It's just a little complex because there's 3 or 4 genes that they're studying. What I wanted to start with today is I wanted to go through another 3-point cross with you, just slowly, just to practice, okay? Remember, these are primarily historical because we don't use this for gene mapping anymore, but it was a way to figure out the order of chromosomes, the order of genes on a chromosome, as well as the relative distance between those genes. Okay, that's those map units, those numbers that we actually calculate. Now I'm going to give you a problem set, and then we are going to go through the whole thing of reordering it, figuring out which one's in the middle, and then calculating it. I'm going to try to do it a little bit better job than I did the last time. Okay, so 3 traits. Like I said, I think this is in corn. Brown, which is designated as BM because it's for brown midrib. So I think that means on the cob. I don't know. That's one characteristic. So this is brown midrib. B for fluorescent seedling. So I think it means shiny. Okay? And PR for purple. Aileron. I don't know what that means. Kernel, I don't know, but that's what we got. So 3 traits, okay? So remember, when we're studying a 3-point cross, we're always going to use a heterozygous female or we're going to use a homozygous female and a heterozygous male. If the male only has one, like in the fruit flies, the male only has one characteristic or trait. So we always start, it will help maybe if I start with the designations. So those are the wild type versions of those 3 alleles. When I put the plus sign up like that, that means it's not the mutant.
Can you say wild type is that the heterozygous?
Well, the wild type means doesn't show, it's whatever color is opposite of brown, iridescent, and purple. These are all recessive mutations. Notice how they're all lowercase. Okay, so they're all lowercase. So they're all recessives. You might get a problem where one of them is a dominant trait, and then that will show instead of the wild type, right? But you signify it the same. If it's a dominant trait, you would use the capital letters, and the wild type, you'd put a plus, right? And then the mutant is the mutant, whatever it is. So I'm going to draw my chromosomes here. I have my heterozygote, so they're carrying the mutations silently, right? We see the dominant traits, whatever those are, not brown, not fluorescent, and not purple. And we are going to cross that corn plant with another plant that is homozygous recessive, a test cross, homozygous recessive. So homozygous recessive, they would have to be this, right? Let's forget about the male-female thing, right? So that really only applies to the flies. Doesn't really apply to corn because it's corn. All right, so we have our heterozygote and we're crossing it with our homozygous recessive. And now we want to know, what do the babies look like? Okay, and we're going to see a parental type or a non-crossover type, and that parental type is going to be the wild type, and obviously this one, because that's the other parent. Go ahead, ask. You're good? You all right? Okay. All right. we don't know yet if this is the right order until we see how many of each of the offspring we collect. Okay, this is just the order they gave us in the problem. And how that usually reads is the problem will say, in corn, brown midrib is recessive to wild type, virescent seedling is recessive, and purple is recessive. That's how the problem starts, right? All right. Yes, no, right? We're okay so far. So we've got the 3 traits. We have the pretend parents because we don't know yet which allele is in the middle. And now we're going to look at the numbers that were collected. Okay. So the numbers that were collected were purple plus the BM 230, purple V plus BM plus 237, PR plus E plus BM 82, PR V BM plus 79, PR plus V 200. Hang on to that one is PR plus BM 195. And then the small ones are PR V BM 44 and PR plus V plus BM plus 42. I'm going to give you a minute to copy those, right?
It's 44.
44, yeah, sorry. It's a 4, yeah. Okay, so now to help you mentally through this process, right? So if this is the correct order of the traits, which of the groups has the highest number of offspring? Not a trick question. The one at the top, right? Right. So if this has the highest number of offspring and represents the order of the alleles in the parent, what would, if this was correct, what would the double crossover be? Right, this one. If this order is correct, do you see the double crossover would be I'd flip the 2 in the middle, right? It's the hypothetical. Okay, I'm looking at what has the highest number of offspring, and I'm just asking the question, is this the correct order of the traits? If as printed, this is the correct order, the double crossover should be the V's flipping position, right? But when I look at the lowest numbers, that isn't it. Right? So that tells me V's not in the middle. You need to say that again, right? So if I'm looking at these as the parents, because they're the most common phenotypes, that means they didn't recombine at all, and they're just the most common in the set of offspring. I'm looking at these, I'm saying, okay, if this is the correct order, if V is in the middle, then I would have this, right? I would get this switch. And then I would get this switch. I would get PR, V, VM plus. And so that's an if. That's one possibility. Then I look to see if the double crossovers match that, and they don't. So I know that's not the right order. I have to rearrange them. Okay, if the double crossover doesn't match that pattern, which it doesn't, then they're not in the right order and I have to fix it. Eraser if that's possible. So I want to stay where we can see, right? So I'm just erasing this and I'm going to put the correct order under here after we figure out what it is. So now let's look at this smallest number of offspring to try to determine who's in the middle, which allele is in the middle. So, and we know these are the parents. Let me erase this double crossover. So what order of alleles can I get from here to here? Which of them is going to be in the middle? Which one is going to be all by themselves? It's not the V or the B because there's the brown, they're staying together, right? And it's not the V or the brown because they're staying together. What has flipped spots? Purple. So that makes purple in the middle. Okay, so when you look at the parents, wild, mutant, mutant, and now we have mutant, mutant, mutant. And this one is wild, mutant, mutant, and we have wild, wild, wild. That means that the purples switched places, so they must be the trait in the middle. So now I'm going to rewrite all of those in the right order, because then it'll help me figure out very easily the distance between brown and purple to virescent. All right, so purple is in the middle. So now let's reorder them. And I'm going to reorder them. I'm going to switch the virescent and the purple. So I'm going to put V first, V, PR, VM. Okay. So now my parents, I'm going to just literally, let's see, I do have enough space here. Let's reorder these. V, PR plus VM, 230, V, PR, VM plus V plus VM, 237. I would go through and rewrite each one of them carefully. Okay. last one, V, P, R, M is 44, and V plus, P, R plus, E, M plus is 42. Okay. There, now I have them all rewritten. And then we want to stop looking at these, right? Because they're not in the right order. I'm just checking if it makes sense with what I have, right? So V, B, R, B, M, I just wrote them backwards, of course. So if I put the wrong numbers, which looks like I might have, V plus PR plus BM 82, no, that's right. Okay, so now we can look at the the non-recombinants. These are the parents, these are the doubles. So the double had to be this, right? Between the parents, we had to go down, up, and the double crossover was like that. To get us to here, we switched the PR trait. So how do I find the distance between V to PR? Which of these shows me the crossover of switching the V's? This first set of numbers or the second set of numbers? I want to know where the crossover is from V to PR and staying down here, right? So V, PR, BM, which would have been this one, V, PR, BM, right? And I'm swapping out the V this time, right? So it was like this, but it was wild type purple, wild type. Now it's going to be wild type, brown, wild type, brown. So we've switched it. We've gone like this. That's the crossover. This first one, we've switched the V's. Where we've switched the V's in relation to the purple, that's our distance from V to purple. So how do we actually calculate that? You guys remember, that's the easy part, right? We take the number of single crossovers plus the numbers of double crossovers, and we divide it by the total number, right? The total number I have is 1,109. And I'll just tell you the numbers so you don't have to calculate them. It was 247 over 1,109. So the map distance was 22.3. Right, so now the other set of numbers are going to tell us the distance between PR and BM, because we're looking at the crossover between, compared to the parents, right? The parents were this, V, PR plus BM, V plus PR, brown. Right, now we're looking to see where these two traits switched, what single crossover gave us that switch. So the V and the purple, so viridescent mutation, wild type, whatever non-purple is, and wild type. So this cross, and we're switching here. So we have the wild type, purple, wild type. So we've swapped those two. That's going to tell us the distance between PR and BM. So when we add those numbers up together, PR, BM. Again, I'll do the math for you. 200 plus 195 plus the double crossovers. We always add those in, divided by 1,109. That gives us a map distance of 43.4 map units. So then we draw our map. here. The total map distance, the distance between VR and purple is our 22.3. Between purple, I should have made that longer, twice as long, right? Let's move purple over closer here. Put purple there. 22.3, and this looks longer. This will be the 43.4, and then that total distance, although it's relative, we would add them up and it's 65.7 for the total distance between V to BM. Iridescent to brown. So the question on the test will ask you most likely, which of the characteristics is, which of the mutations is in the middle, right? So what's the gene order is what it'll say. Hint, which one's in the middle. And then what are the map distances between these three traits? Right? Okay, so try the other ones that I put on the board, that I put up there for you, right? And then we can talk about it again if we need to on Thursday.
How did we, I just wanted to discuss, how did we, find out that, the V was the middle,
So by looking at
Yeah.
what the parents are,
Mhm.
and then looking at the double crossover. The double crossover reveals which characteristic is in the middle, because in that double crossover, one of the traits is going to flip positions relative to the parents. Okay, so in this order, V does not switch positions to get us to these double crossovers. So therefore, the order is not correct. And we need to change the order. So we find the trait that when it flipped from the middle gives us these numbers, the lowest numbers. And in our scenario, right, so we have this, so we have B. Remember, one of the parents is heterozygous, right? And the other parent is homozygous recessive. So we're actually, you can kind of shorthand it to just looking at these two chromosomes because they represent both of the parents actually. So the only way that I get this combination, B plus PR, BM plus, so wild or purple phenotype alone, and virescent non-purple and brown, non-brown. Only way I got that is, oh wait, I'm going the wrong way. This plain purple plain. The only way I get this is if purple's in the middle, and that's the one that gives me the lowest numbers in the double crossover. So I change the order so that purple's in the middle. Some people don't bother, but I like to do it because then it helps me figure out which is the distance, which are the numbers I'm using for the distance from V to PR and from PR to BM, right? Because the distance from V to PR is where we're switching out the first characteristic, and the distance from PR to BM is where we're switching out that third characteristic. So practice one more, right? It'll be one problem on the test. Practice one more. And then I have one more in my pocket in case 14 or 15 doesn't go well. All right, and then that's it. We're gonna leave chapter 5 now. Yay. And we're gonna start chapter 6. Start something new. chapter 6 is exciting for a couple of reasons. Again, unfortunately for you, it's mostly historical, but we're actually going to do some of the labs that are associated with chapter 6. So you will get a little experience, and some of the techniques are still in practice in the laboratory. Okay. So we're going to start with, this is called mapping in bacteria and bacteriophages. How many of you have already had microbiology, like completed it? Yeah, we don't actually offer microbiology too, so that's like a pretend class. So yes, most of you. So this will be familiar to you, okay? But because there are some people who haven't gone through it, it might be boring for you, right? So we're going to go through this chapter. It would have been basically part of your chapter 9 if you took micro here in that in Cohen in the book. So we need to talk a little bit about bacteria first, again, for those of you that are not finished with microbiology. And then we're going to talk about ways that bacteria naturally can move some of their genetic material between cells. Okay, and there's 3 major ways. Again, I'll just lay this out there for those of you that are familiar. Conjugation, transformation, transduction. It's basically what we're going to be talking about. And then in class, we're going to do a transformation experiment. We're going to move the jellyfish glow gene from a plasmid into a bacteria, and then the bacteria are going to glow fluorescent. And then maybe we're going to be able to make those bacteria make enough of that protein that we can run a Western blot and actually separate that protein from all the other proteins the bacteria is making and see it on one of those flat electrophoretic gels, but we'll see. So I would remind you that all the bacteria are prokaryotes, right? Which makes them convenient to study in molecular biology because we don't have a nucleus. We don't have to blow it apart. We don't have to treat it extra hard. You can basically put them in a little dish soap and shake them up, right? And they open up and you can get their DNA out. The DNA is a single circular chromosome, right? So we only have 1 chromosome to worry about. It does not have a lot of histones wrapped around it, or it's not wrapped around a lot of histones, I should say. So eukaryotic DNA has, with all that DNA, it has to be packaged up somehow so it'll fit inside the nucleus. In bacteria, it's just laying all over the place, right? It doesn't look like a circle, but if we exploded it out of the bacteria and stretched it out, it would make this giant circle, okay? So 1 chromosome, circular. And why that's important is because that does give it a little more stability than if it is already in pieces. Our favorite bacteria to use in molecular biology in genetics is E. coli, various strains of E. coli. It's our favorite. We call it our lab rat of molecular biology. And the E stands for Escherichia coli. It is a gram-negative bacillus. It is non-flagellated most of the time. Okay, so it doesn't move. It's rod-shaped. It's gram-negative, so it doesn't have that super thick layer of peptidoglycan around it. It has a phospholipid bilayer, a little bit of a cell wall, and another phospholipid bilayer. So in a lot of ways, it's easier to get things in and out of this kind of a cell. It grows super fast and will eat just about anything, right? So we say it's a ubiquitous organism because it's not fussy. We would say fastidious if it's fussy. Like if you want to grow, you know, I don't know, I'm trying to think of a fastidious organism. If you, I can't think of any. So we'll just skip it. So it reproduces, it has a generation time of about 20 minutes. 20 minutes.
So making more generation every 20 minutes.
It makes makes a copy of itself every 20 minutes. So if I start with one single E. coli cell, which I can never do because they live in a pile, right? You've seen bacterial colonies, they live in a clump. But if I could start with one cell, right? After 20 minutes, I'd have 2. After 40 minutes, each of those would reproduce, I'd have 4, right? So it's a geometric progression, not arithmetic. At the end of 8 hours, I have over 1 million cells, over 1 million. At the end of a whole day of 24 hours, I have a number followed by 23 zeros, right? That's a lot of offspring, right? In one 24-hour period. And they're small, obviously. I can grow them in broth. So I can grow them in a liquid media. I can put them in a flask and put them in a shaking water bath to keep them warm, give them lots of food, and like literally come back tomorrow and have billions and billions of them. Or I can grow them on solid media that we call agar. If I need to store them, if I need to pick an individual colony that's not touching any others, if I need to isolate them. So that's awesome. And their nutrition requirements, like I said, are very minimum. You can grow them in, we usually grow them in what's called LB broth. It's actually called LB or LB agar. It stands for Luria broth, named after a very famous early molecular biologist, Salvatore Luria. And because this bacteria only has this one circular chromosome, people have mapped the genes on that chromosome, characteristics of E. coli, believe it or not, and they've mapped approximately 6,000 different genes or characteristics. So it isn't like we're trying to study 2 things, right? And in a human, for example, we have between 25 and 40,000 working genes. That's kind of a much bigger project, right? Like I talked about the last time when that woman stood up and said she's been assigned chromosome 2, right? I was just like, "Ooh, you'll be doing that the rest of your life," right? So it's a lot more manageable. We only have these 6,000 genes, and many of them you can see expressed in a different phenotype. And I know that might be hard for you to imagine, but the size of the colony, the edge, remember all the characteristics that you had to look at in microbiology, right? How big is the colony? What color is it? What does the edge of the colony look like? If it grows in broth, does it grow faster or slower? If I feed it this, will it produce an acid and change my media a different color? So there are lots of known ways to manipulate this organism, right? Okay, now there are some pictures of some bacteria. So we've mentioned that. So let me say this. So this generation time of every 20 minutes gives us this growth curve that we refer to as the bacterial growth curve. And it's relevant because you can very quickly estimate to a pretty close approximation how many cells you have at different periods of time, because so many people have grown E. coli in culture in a water bath, starting with this many cells or approximately this concentration of cells, let it grow for an hour and then looked at it. So this, we call it the bacterial growth curve. And it's always the same curve. Time in hours is on the bottom, and along the side is log number of bacteria. And the curve always looks like this, approximately. The first part of the curve we call the lag, LAG phase, because that's the first hour or 2, maybe. I'll put this at 0, so I'll put this at, let's make it 2, 4, 6, in 2-hour increments. So that first 2 hours, you're not getting a lot of doubling. You're not getting maximum growth. Then there's a huge spike, and this spike is called the log phase or the exponential phase. And this is important, or I wouldn't be bothering to tell you this, right? This part where the peak flattens off, we call that the stationary phase. And then this is the death phase, right? The bacteria are doing all of their metabolism. They're expressing all of their proteins. They're making all of their enzymes. They're going to respond the best to an antibiotic. You can kill them the best in this exponential phase here. That's the sweet spot. So as a scientist, if you're using E. coli in your laboratory or a particular strain of E. coli, you need to know what that sweet spot is, because that's where you're going to actually conduct your experiments or collect most of your data. Right? So you need to know what that time frame is. Is it really between hour 4 and hour 8? Or in my laboratory, is it really between hours 2 and 4.5? Right? And how the scientist knows this is what they would be doing is they would be dipping into the solution every 30 minutes and counting how many bacteria there are per milliliter, right? We call them, and then usually actually they would grow them on a petri dish and they would call them colony forming units per milliliter. And you count them, and then you know how many cells you have per mil. And again, the object of the game is to get to the sweet spot and to have it match your procedure, right? So if I need 100,000 E. coli cells to do this experiment, again, I need to know what time of day I find that number. So as I mentioned, what you can do is suck a little bit of solution out and actually sit there and count how many cells you have under a microscope. Or you can do it the easy way, which is you can use a spectrophotometer. Do you use those in chemistry? Yes, Dr. Stewart, we've seen a spectrophotometer. No? You can get an approximate count by measuring turbidity of the solution. And I do that using a spectrophotometer. This is a really powerful tool in molecular biology. We use it all the time. We use it to quantitate how much DNA we have, how much RNA we have in a solution, how many bacteria we have. And so all laboratories will have various forms of a spectrophotometer. Okay. Some laboratories now use the spec to quantitate the DNA before they put it into a PCR reaction, especially if they're using a qPCR, which is a real-time PCR reaction. All the quantities have to be in the same neighborhood so that you get the best binding of your primers, or it doesn't work very well. Okay, so we have some ancient Greece spectrophotometers in there. They're called Spec 20s. They're for student use. We can take a look at them later. And we probably aren't going to go to the trouble to use them because we're using kits, so everything's already been optimized for us. But if we were trying to do this experiment ourselves from scratch, we would have to do this. So what the spectrophotometer does is it measures, it just compares, basically, right? So I have a light bulb. And I'll just tell you, it's usually a UV light bulb. We usually measure the turbidity at 260 or 280 wavelength. So it's usually a UV light bulb, but I'm just going to draw it like a regular light bulb. And what we're measuring is how much of the light gets through our glass tube. And it hits a detector, and I'm drawing like, you know, a Flintstones detector here, right? Like one from the Stone Age. And then that detector is going to read for you either absorbance or turbidity, or T. 100% turbidity is equal to 0 absorbance, right? So they're marked like A/T on the machine. So if there is just froth in my tube and all the light from the light bulb hits my detector, my little needle goes over here to 100% transmission. Right, so 100% T, right, equals 0 absorbance. The light is not absorbed at all. It's all going straight through there. And again, this is like super simplified. Now, if I take that tube out of my machine and into it, I put a tube that has bacteria that are growing in it at some density. I don't know, maybe it's at 30 minutes, maybe it's at an hour. Now look what happens to the light rays when they come off of here. If they run into a bacteria, whoops, they're going to be reflected and refracted. They are going to be bent. So now, look, not as much of the light is actually going straight through and hitting my detector. So I did 4 lines, so I'm going to do 2 lines on this one. So now my absorbance, or my percent transmission, has gone down to 50%. Again, this is super crude. What someone has done now, before most of you were born, is then they sampled this tube at 1 hour that was 50% T, and they actually counted how many bacteria were in it and said, okay, that's approximately 55,000 E. coli cells per microliter or per milliliter, whatever. Right? And they continued to do this for the whole time to map out where this curve is. No, you're just like, who would do that? Because then once you do that, you have a shortcut. Now, at any time of day, I can take a sample, put it in my spectrophotometer, and whatever absorbance I get corresponds to my graph. And I'm like, okay, I have enough cells or I don't have enough cells. So I'm going to go to lunch or I'm going to get started right now. Right? Notice that what comes after exponential is stationary phase. That means as many cells are dying as new cells are reproducing. That's no good. Now there's just a bunch of junk in my sample, right? It's a bunch of excess acid, a bunch of broken up bits of bacteria that I may not want as part of my experiment. And then of course, when I get to the death phase, more of them are dying than I'm getting new cells. That's no good for me if I'm trying to optimize using my bacteria when they're growing best. Okay. Again, for our purposes, our kits have all been optimized for us. So we're just going to go with what it tells us to do. Incubate this for 2 hours, incubate this for 4 hours, incubate this overnight. You should have enough cells, and we're just going to give it a whirl. Okay, because you're not actually doing like a quantitative scientific experiment, right? You're not actually doing qPCR on these bacteria. Okay, now here are some petri dishes. They show you some petri dishes with bacteria growing on them in case you've never seen that. The bacterial growth curve is in there. All right, now on to the mapping. We'll start with conjugation first. So because bacteria, some bacteria, in addition to their chromosome, have a plasmid. And a plasmid is extra DNA that's not part of the chromosome. We call it extra chromosomal DNA. It is also a circle. It's a lot smaller, carries a lot fewer genes, maybe a couple hundred. I like to tell, like my microbiology students, the first thing I say to them is plasmids usually contain bonus genes. And what I mean by that are non-essential genes. If I'm an E. coli and I've got my 6,000 genes, I'm living my E. coli life, right? I don't really need these genes, but if I've acquired them, and let's say on the plasmid is a gene for ampicillin resistance, that's good for me as the E. coli, bad for you if you're my host, right? Because now if you take ampicillin, I have the enzyme that destroys ampicillin and I'm resistant to that particular drug. Or maybe it's a gene that I can make an extension like a fimbriae. So now I can stick to things better. Or maybe it's a gene now I can make a capsule so I can stick to things better. I didn't have that before. I was doing okay, but now that I have it, I'm doing even better. I'm multiplying even more, making more of an infection, right? So these are non-essential genes, but they usually give the bacteria extra characteristics. Might be a toxin. Oh, now I make this toxin that gives you just like wicked bloody diarrhea that you can die from. Okay, it's good for me as the bacteria, bad for you as the host, right? So extra chromosomal, bonus genes, non-essential genes, but usually they give the bacteria a new characteristic or trait. Where do they come from? Well, they come from other bacteria, okay? And once a bacteria has one, in addition to the bonus genes on that plasmid are the genes to construct a pilus or a little tube that I can stretch over to anybody I'm touching, and I can give them a copy of my plasmid. Okay. We call that conjugation, that process, transmitting have 2 T's or 1?
Transmitted has one.
1, just 1? Transmitting to a new E. coli, new cell. And it's important that you see the word copy. So I'm not just giving away the one that I have. I'm keeping the one I have. I'm making a copy and sending it over to my friends. So, for example,
You're right.
2 T's. If one of you had a copy of the exam from last week and thought it would be a good idea to share it with everyone that you had there, everyone in the group chat in genetics, right? There, that's the same kind of thing, right? You would share it and she would share it. And you're like, oh, I know somebody at the other table, so I'm going to share it. And pretty soon the whole population has this characteristic, right? Now there's words, vocabulary that go with this process, that go with this. Why are we talking about this, right? Because the genes on that plasmid are always in the same order. So when they are transmitted, when they are copied and given to my friend, they always go in the same order over to my friend. And if the connection between us gets broken and my neighboring plasmid friend only gets 2 of the genes, and a different friend that I started to copy gets 3 of the genes, and a different friend gets 6. You see how just like we had the flies and the plants with 2 and 3 characteristics, we can start to map what the order of the genes are based on how long the plasmids were allowed to copy. Okay, so that's where this is going. You can map the genes on the plasmid in the order based on time experiments where you disrupt on purpose, little shake or maybe a little blender, right? Where you disrupt the conjugation and then you look to see the new bacteria that were getting the plasmid, how many of them got this trait, how many of them got that trait, how many got this trait, how many got this trait. And then you can start to map the order of those genes based on time. Okay, so like I said, there's words to go with this, lots of words, right? We call the bacteria that has the plasmid. So this is the, I'm going to tip E. coli right side up now. This is the plasmid down here, right? This is the chromosome. This is the plasmid. We call the bacteria that has the plasmid an F plus cell because they called it the factor, basically, the F factor. And one of the genes that is always on the plasmid is this ability to make a pilus, make proteins that allow it to make an extension, a tube that's going to attach between the neighboring E. coli cell. So here's my neighboring E. coli cell with its chromosome. This cell is going to be the F minus cell because he doesn't have a plasmid. The F plus cell, you might also see it called the donor cell, and the F minus the recipient. So always one of the genes on the F plus on the plasmid is this ability to make an extension that we call a pilus. It's made of pillin, the protein pillin, same protein, very similar protein that if an organism makes fimbriae, makes lots of them. We call it a pilus if there's only a few of them. So pili, right, would be, or pilus is the singular, pili is the plural, right? So let's say this bacteria is also has one going over here to a different friend. Yeah, I should have taken Latin sometime in my life, but I didn't. So, right, then we would call it pili, right, because there's multiple of them. And sometimes you'll actually see people call them, because this is known as one of the ways of horizontal gene transfer. People call it bacterial sex, or they call this a sex pilus. So you might see that as well. Conjugation is one form of horizontal gene transfer. Told you there were a lot of words to go with, right? I think it's good for you to get the big picture first and then like learn all the words, but whatever. So time lapse, right? My first E. coli cell that has the plasmid produced this pilus, this sex pilus, to its neighbor, and it says, hey, I'm sending you a copy of all the genes that I have on my plasmid. That's cool. And like I said, depending on how long the time that these cells are connected, that's going to determine how many of those genes get over there. They always go in the same order because the site to replicate that plasmid is always in the same place. It's always the same gene sequence. So they always start with whatever that first gene is. They always send over in the same order. And then we're going to be able to map which one came first based on time studies of disrupting this sex pilus. Once E. coli 2 here gets a full copy of the plasmid, he's now converted to an F plus cell. Okay. You'll see a lovely picture of this on page 128 in your book. And it's not rocket science. They would give you like a problem of this. They would give you, this is the amount of time that these different characteristics took to show up in the second generation. And then you would look to see which one was always present. And that's the one that went over first. Which one is present in the next most of the cells? That one is second. Which one is the least present? That one is the one at the end. right? This is a very famous map because this is the one people did first. And obviously, it's a lot easier for us to just talk about it than it was for people to design experiments to actually do this. But let's see if I can show this to you. And then maybe. Bacterial cells do this all the time. But it's not really like, it doesn't matter to us, of course, unless it's something that we're concerned about as far as giving bacteria a characteristic or resistance to an antibiotic that we don't want them to have, right? Please. Bigger. Okay. Now, occasionally, I know this says it's a different word. Occasionally, when that chromosome, that plasmid comes over, sometimes the cell converts to what we call an HFR cell, which stands for high frequency recombinant. And in that case, the plasmid has incorporated itself into the actual chromosome. And now it's a permanent part of that organism. So that's why they're showing HFR strains. But let's just, these are some strains, order of transfer, right? Earliest through the latest, we're looking at 1, 2, 3, 4, 5, 6, 7, 8 characteristics. And literally it's based on time. And that's how we designate the places or locations on a bacterial chromosome. We designate them in minutes because the chromosome's a circle, like a clock, which is a familiar thing to us. And so obviously there's only one chromosome, so we can't say short arm of chromosome 2, right? So we say, oh, the lac gene is located at 10 minutes. So that means 10 minutes from the origin of replication or the start of where the plasmid would have grabbed this gene and taken it over to another cell. So let's look at this. Actually, I'm going to change my mind and show you this instead. This is what I was talking about the timing, right? So let's just look at this. So minutes of conjugation, and then they disrupted the sex pilus in the experiment after 10 minutes, 15 minutes, 20 minutes, 25 minutes. Which gene is going to be first on that plasmid? Which gene comes over every time and appears first? The blue one, that's AZI. It's resistance to an antibiotic. See how it says a capital R next to it? That's a resistance gene. And then which gene is second? Yep, the green one. There's the lac gene. Looks like it's at 20 minutes, not 10. I was wrong. And then the galactose, the galactosidase gene looks like it's at 25 minutes. Right. So it's, like I said, it's not super complicated, but in practice to actually try to do that is a lot more complicated than just looking at the results. Right. Okay. What time is it? All right, we're going to stop now. And we're going to finish that lab, and I want to give you back your exams. And then Thursday, if there's time, we'll start making some buffer at the very least.